Hi, everybody. Hopefully you can see my screen now.
We can see and hear you. Thanks.
Great, great. So hi, everyone, and thank you all for joining. It's great to see such a large number of people attending, so it's very exciting. It's a topic that I think is very important for the science community as a whole. It's certainly important for my lab, and I'll touch on that in this talk. So my name is Dan Beiting, and I'm an assistant professor of pathobiology at the Veterinary School at the University of Pennsylvania. If you're interested in the kind of work we do, you can check out our website because in addition to having a research lab, I help direct a center for host microbial interactions. And so broadly speaking, my research, which I really won't get into the details of my research in today, but my research deals with infectious diseases and how we can use cutting-edge genetic and genomic tools to understand how pathogens interact with their host. And so one consequence of the kind of research we do is that we generate a lot of data and we use a lot of software. And so we often find ourselves feeling a bit like this, or in this position where we're sort of surrounded by our data outputs, right? So we have code that we write or that we reuse from other people to help us analyze large amounts of data. And these large amounts of data are coming out of the kinds of experiments that we and many other labs are doing. This is a major research product that is ubiquitous across biomedical research, large data generation. And then, by combining our code with our data, we generate various visualizations of our data, and we kind of iterate through this, looking at our data in different ways, slicing our data in different ways. And so this really poses a real challenge, not just to myself as a lab head, but to anyone in biomedical research, right? Because at some point, we have to kind of take this confusing world that we sit in, and we have to communicate it in a clear and concise way to our supervisor. And we have to communicate it in a clear and concise way to our team. And I'm using that term very generally because that team sometimes might be your lab mates. Sometimes it might be an audience at a scientific conference. And other times, as we're seeing now with the COVID-19 outbreak, it may be the public, right? And I will say there's another important person in this team, and it's yourself, right? So the other thing we really struggle with in science is knowing exactly what we did 6 months ago and how to do it again in a way that's efficient and accurate, and how we can sort of pass off the research we're doing to a new researcher that might be coming into the lab in a way that they can pick up and start doing that work just as efficiently as you were doing it when you sort of passed the baton to them. So that's really the challenge. And I think whether you're talking about protocols.io or CodeOcean or other tools, we're looking for solutions that help us in this space, because communicating the results of our research products is really challenging to do. So I think, ideally, we'd like to be moving toward the right-hand side of this reproducibility spectrum. And this is quite old now. It was published by Roger Peng almost 10 years ago, but I think it's very relevant and topical today. And unfortunately, I think most laboratories, most researchers still sit on this far left end of not being very reproducible. Journals have tried to help with that in implementing strict guidelines for how we make our data available and how we make our code available. But by and large, we focus still on publication, right? So my sort of feeling, and I think, one of the big challenges for biomedical researchers now is getting folks on board with this. But my feeling is that if you start in a research project actually with the intention to operate on this end of the spectrum, then you strive for the highest quality of work. So if your goal at the outset of a project is to be able to produce a research product that is reproducible, that's shareable, that's findable, and that can sort of lend itself to full replication, then by its very nature, then you're trying to make a project that's as high quality as possible, because people are going to be able to look at that project in great detail and reproduce it. So I'll show you an example of how we've tried to move from the left to the right in this reproducibility spectrum, starting with this idea of jumping from publication only to putting together publication and code. And I'll give you examples along the way of what we're doing. And by no means are we experts in this, but I think we're getting better. And it takes practice, actually, to get really good at this sort of thing. So publication and code. Well, one way we've tried to do that is we always include a supplementary file in our papers. And this is an example of a Science Translational Medicine paper that I'll bring up again in just a moment. But, you know, attached to that paper is a PDF file. And that PDF file, and the details of this aren't so important, but that PDF file, when you open it and view it, has a very organized table of contents that walks you through our research logic, basically. How did we tackle this problem, and what's the code that we used? And how did that code link to the actual graph or plot that we produced? So I've highlighted in red text here. They can simply click on this table of contents, and they'll be taken to an explanation of that figure, the code that was used to generate it, and the figure itself. And we've done that in other cases as well, even when we're not the ones leading the work. So, you know, here's an example of a lab also at the veterinary school that I work closely with. And this was not our work, this was their work, but we helped them analyze the data, and we encouraged them to produce the same sort of output, right? So a supplementary code file that's organized and easily accessible and transparent. So this really helps us with transparency in our research by bundling together our publication with our code. We've now, you know, moved towards putting together not just the paper, but the code and the data, right? So that PDF, when people open it, there's no data there. They have the code, they have the output, but they don't have the input. So how do we give them the input? Well, here's an example of a paper we published last year where that paper is linked to a GitHub site. And if you're not familiar with GitHub, it's not a big deal. It's a collaborative software development platform. It's free. But many people use it just to house their scripts and data and link it to a paper, okay? And so these are called repos or repositories. Here's the GitHub repo for this study published in Microbiome, and it has a detailed explanation of what you can find there and how to take full advantage of it. But having this GitHub repo in no way sort of provides an immutable connection between the paper and the code and the data. And so we take this a step further, and we use something called Zenodo that lets us put together our GitHub repo with data and to tie that to our paper and give both our paper, of course, the publisher does this on the paper side, but on the data and code side, we get a DOI for that Zenodo implementation as well. All right, so now this is sort of immutably linked to the paper. If we go in and change the GitHub repository, it does not change this Zenodo. This is sort of time-stamped and locked down. And now this is, I think, a better approach to gather all these components together. But it's not the final sort of step in that reproducibility spectrum, because it still has problems. And those problems are that there's really, right, the obvious one, right? There's no computer here. So anyone who were to access this paper, the GitHub repo, or the Zenodo resource would not have a computer to run that code and produce that research output again. But I mean, maybe more relevant is that they don't have the software. I just told you in the beginning that we use a lot of bioinformatics code, but also a lot of software written by other people to do our analysis. And none of that software is included in any three of these pieces that we've put together here. And that's actually a real challenge, because installing bioinformatics software requires that you work in the command line, sort of this, working under the hood in your computer. And a lot of times getting the software installed is really challenging, because the software that we used changes by the time you go and try to install it 6 months or a year from now. So that really raises questions about, is this really reproducible? If the individuals who look at these three components here can't actually redo the analysis, then it's clearly lacking something. So the nice thing is we're in a time when there are solutions to all of these problems, but never, at least not until we sort of came across CodeOcean, never really in one place. So the solution for not having hardware is that you use a cloud computer, right? You use a computer that's been set up by Amazon or Google Cloud, and you access that computer through a web browser. The problem of not having software, also an easy fix. There are solutions that bundle together many different pieces of software, programs, et cetera, into units that make it easy to install them. And these are things like Docker and other resources that let you easily bundle up software. And then if you have a cloud computer and you have an easy way to get software onto that cloud computer, then you're in a position to run a full analysis on the cloud. If you can connect that cloud computer with these research components. So that then brings us to this idea of what's really needed for full replication. How do we link our code, our data, and our publication in a way that anyone, regardless of their bioinformatics expertise or their skill level or knowledge, could reproduce the data, the results you got in that paper? And so that's where my group really started becoming very interested in CodeOcean and worked closely with the great team at CodeOcean to help get us started and get off the ground. And CodeOcean is really that solution that I mentioned that kind of brings together this idea of cloud computing, easy installation of software, and the ability to then take this cloud instance and anyone can reuse it, copy it, and then modify it. And so I thought I'd give you an example of that. Just going to go over to my web browser here. So if you click on the link in the bottom of that slide, it takes us over here to CodeOcean. And what you're looking at here is one of these code capsules that CodeOcean produces that bundles everything together. And so on the left-hand side are all these components, these inputs, data, code. And if I click on my environment button, I can see that here I have all my software. So I have my data, I have my code, I have my software, and I have an operating system. I have a computer, all in the cloud, all accessible through a web browser. Now I don't need to worry about installing software. It's already embedded in this code capsule, and anyone who publicly, and this is a publicly available code capsule, anybody who discovers this capsule or who gets here by following the link in the manuscript, for example, they find themselves in this exact same environment. This is the public-facing version. It's published, see? So they have everything at their disposal. And if they choose to, and this window's in the way here, they can rerun the entire analysis of the paper. And what you can see is the output that they get is every figure from the paper. So as an example, clicking on any of these will open those figures. So here is one of the sort of a complex graph that we produced for the paper, easily reproduced here, no guesswork involved. Other simpler graphs, also easy to produce, and on, even tables of data, which is shown up here. So the full manuscript is completely reproducible. Okay, so where does that leave us? Well, we were able to take this with CodeOcean's help a step further by actually embedding that code capsule into the manuscript, into the publisher's manuscript web page, so that folks who are reading this paper don't need to go directly to CodeOcean. They can see that that reproducibility is actually embedded directly within the journal page. And so we'll head back to the browser here, and I'll just show you. Here's the journal page. If we just scroll down and we click on supplementary materials, we go all the way down to the bottom. Here is the CodeOcean capsule for this Science Translational Medicine paper embedded right in the journal page. So they could actually run it from here and reproduce what we've done in the paper. All right, so I thought I'd just close by some of the other ways we're thinking of using this kind of platform. and that is to promote reproducibility in the classroom. So we talk a lot about, the idea of reproducibility, and usually when we're talking about reproducibility, we really mean with respect to data and analysis and the scientific method, but I feel there's another need for reproducibility, which is in teaching. And if that's never been more clear than now, where we have this massive need for online, for students, graduate students, undergraduates, to tap into online learning materials that are delivered in a consistent and clear way. And so we have a course we've been running for about 5 or 6 years on analysis of gene expression data. So again, it has a biomedical slant to it, of course, because that's our lab's interest. But it deals with a lot of the same issues many people do in teaching graduate students in biomedical sciences, which is how to analyze data, how to think about it, and how to work with large data sets. And so, really what we want then is this idea of FAIR, or the idea we usually apply to our data, making it findable, making it accessible, interoperable, and reusable. We'd like those FAIR principles to be applied to our teaching as well. And so what we've done now is we've connected our classroom to make it completely reproducible, the actual class itself. And the way we do that is our entire course, first of all, is hosted on GitHub. Even the course webpage is served by GitHub. So it can be copied in its entirety, all the content of the course, the syllabus, the data, the scripts, even the links to our videos on Vimeo are all easily copied. All someone has to do is just copy our GitHub repository for the course. In addition, through CodeOcean's help, we were able to set up a course capsule. And so this capsule now, instead of embedding all the data and all the code for reproducing an analysis in a paper, it bundles all the code and all the data, all the homework for a course. And so I'll just head over again to the browser, and this is sort of end of what I have to show you. But here is that course capsule. Now we don't have to spend time up front having students install software. It's already installed. Their computer is already set up. And we have all of the steps of the course broken down here for the code. We call these hack dashes. This is homework, in-class working projects. We have those all here. We have data, and CodeOcean, because of the way they work, they're able to load publicly available data sets so that they're accessible to anyone who copies the capsule. So we have data, not actually part of the capsule per se, it's being pulled from a public data repository that CodeOcean has set up. So the students have a lot of data sets to work with in the course. They have data sets I've provided as well. And then they have all the code, all the software, and the computer. So when teachers are looking for materials they want to use to deliver as online curriculum, they could simply copy this course capsule, and they could have everything they need. They could also copy the website through GitHub. Okay, so that's really it. I'd be happy to take any questions you might have, but that's really how my group's working on improving our transparency and reproducibility, both in our research work and in our teaching work.